Good day. I'd like to welcome you to the 28th commencement for the Skaggs Graduate School in Chemical and Biological Sciences. My name is Jamie Williamson. I'm the Executive Vice President for Research and Academic Affairs here at Scripps Research. I'm standing here in the auditorium alone. We're having virtual graduation this year. The coronavirus has really knocked us for a loop in 2020, and I think 2020 will be a year that we all want to forget. But I don't want the coronavirus to diminish the sense of accomplishment by the graduates or the sense of pride by the families who would normally be gathering here with me, cheering them on in a processional to graduate. No trumpets this year. No processional, no speeches, but we were going to come together virtually to celebrate and honor the graduating class. We can't let coronavirus define what's happened this year. Those of you who have graduated will go on and take jobs in the pharmaceutical industry, in the biotechnology industry. And this is why you came to Scripps. You came to suit up for a career in biomedical sciences. So I hope, if anything, this pandemic has served to focus your energy and enthusiasm for the career that you've chosen. And I hope that you have a sense of pride and accomplishment for coming to one of the top graduate programs in the world and being trained here to go suit up for these very challenges that we face in human health today. So please join me in listening to each advisor introduce their student with a special sense of pride about their individual accomplishments. But let me remind you that this class has done an incredible job under incredibly difficult circumstances as we wrapped up this academic year. And all of the families should really just be brimming with pride at the accomplishments of the graduates. So we will continue with the commencement, and it's my pleasure to introduce Dean Phil Dawson.
Thank you, Jamie. I'm Phil Dawson, Dean of Graduate and Postdoctoral Studies. It is an honor to virtually present the doctoral diplomas to the wonderful group of graduates from our campuses in Florida and California. I led the admissions committee when this graduating class applied to the program, and I was proud to see them accept our offer at that time. And I'm even more proud to have watched them grow as individuals and as independent research scientists. While we celebrate the accomplishments of our students, we should also recognize their friends and their families. Thank you for the gift of patience and support that allowed them to focus their time and attention on their research. They could not have done it without you. While much of our focus today is celebrating our graduates, I would like to take a moment to reflect on the memory of someone who is no longer with us. Our Associate Dean, Wendy Havron, passed away earlier this year. I'm privileged to have had the opportunity to learn from such an accomplished and compassionate colleague. I always appreciated her frank and assertive input during our discussions about the graduate program and the role that faculty have in mentoring the next generation of scientists. In addition to her significant contributions in the field of immunology and wound healing, Wendy was honored with the 2018 Outstanding Mentor Award by our Society of Fellows. Wendy made a strong impact on many of our lives, and she will be missed. During this ceremony, our faculty mentors will briefly highlight the individual achievements of our graduates. And for the first time in this ceremony, we will be hearing from the students themselves as they receive their diplomas. Before we start, I would like to take a moment to highlight some of the collective achievements of the graduating class of 2020. While there were many metrics of success, publications are the primary way that we communicate our achievements to the outside world. I have fresh publication data, and I'm pleased to report that the students graduating today have collectively published 293 manuscripts. This is an astonishing output, and I suspect that many of our graduates have a paper or three still in the draft stage. These papers are communicated in the world's most prestigious journals, including Nature, Science, Cell, PNAS, JACS, and the Journal of Virology, Immunity, and Neuron. Many of these journal names have gained public awareness in recent days with the news reporting of advances in COVID-19 research. With an average of over 6 publications per student and counting, it is clear that the class of 2020 has been one of our most productive cohorts in Scripps history. I look forward to watching our students and mentors describe their accomplishments during the 2020 graduation ceremony. I will pass the mic to Pete Schultz, our president.
My name is Peter Schultz. I'm the president and CEO of Scripps Research Institute. And I'd like to start out by also offering my congratulations to the graduating class and their families and friends. I'd also like to introduce our first faculty member, Phil Barron, who's a professor of chemistry, who will lead off the introduction of the 2020 graduating class.
Hi, I'm Phil Barron from the chemistry department on the La Jolla campus, and it's a joy to present Yuzuru Okanda to you today. Yuzuru was born in Yokohama and raised in Miyagi. He obtained his undergraduate education at the University of Tokyo, where he conducted research in the lab of Aichi Nakamura. For his thesis project at Scripps, he chose the most challenging and complex problem in total synthesis our lab has ever embarked on, the two-phase total synthesis of the legendary billion-dollar anti-cancer medicine known as Taxol. Heralded by many as the most complex terpene target out there, his goal was fundamental in nature and sought to answer the question of whether modern organic synthesis could offer a concise path to this target that would be amenable to medicinal chemistry explorations. Targeted for the past 30 years by hundreds of chemists, this natural product has been made 10 times before, only arriving at sub-milligram quantities of material after 40 to 70 steps. In contrast, Taxol is commercially prepared in metric ton quantities through plant cell fermentation methods. While landmark contributions, they stood as a reminder that such complex molecules could only be realistically made by tuning biological systems and running medicinal chemistry campaigns by degradation or semi-synthesis from Taxol itself. Starting 13 years ago, we set out to see if mimicking nature's approach to creating Taxol would deliver a more concise approach that would also be amenable to generating analogs that were previously inaccessible. Yuzuru was the final PhD student to work on this project, and despite everyone in the lab cautioning him against it, he not only took the challenge, he finished it like a samurai warrior through a combination of sheer brilliance and mind-blowing dedication. In nature, no less than 20 different enzymes convert simple hydrocarbon starting materials to Taxol through 2 phases, one which builds the carbon skeleton and one which adorns that skeleton with oxygen atoms in a similar manner to decorating a Christmas tree with ornaments. Yuzuru's route, which is holistically mimicking this approach, involved only 23 steps, requiring only 4 steps to generate the tree and 19 steps to add all the ornaments. Yuzuru is a force of nature, seemingly immune to the emotions encountered by endless failure and lack of sleep. He is internally driven and motivated, and despite his fierce approach to solving problems, he was a great friend to many at Scripps, never too busy to help young students and the life of the party at all gatherings. He is already sorely missed here at Scripps and is off to an amazing start of his postdoctoral stay in the Radosevich lab at MIT. Congratulations, Yuzuru.
Hello, my name is Donna Blackman. I'm professor and chair of the chemistry department at Scripps La Jolla campus. I'm delighted to be here today to celebrate David Hill's accomplishments in completing his PhD. David came to us after graduating summa cum laude from Northeastern University, followed by 2 brief stints doing research at national laboratories. David quickly became a key resource for students and postdocs, not only in our group, but also in other research groups as well, both here at Scripps and with our collaborations outside Scripps. David carried out most of his PhD research with me in collaboration with Jin Chuan Yu's group in the National Science Foundation Center for Selective CH Functionalization. But I guess I didn't give him enough work to do, because not only has David co-authored a number of papers doing this work, he also published with the Angle Group at Scripps. But I want to highlight in particular his first author, Jack's communication, in which he developed a new protocol that uses the enantioselective reaction itself as a marker, dynamic chiral ligand exchange. This is an idea that David came up with on his own, and it makes an important contribution to mechanistic studies in asymmetric catalysis. David is an intrepid traveler. He's always been sought a variety of experiences. Before the coronavirus curtailed such activities, David spent short research visits at Imperial College London and at KAIST in South Korea. He really shines as a collaborative researcher, which is a quality that will serve him very well in his future career. He is currently beginning a postdoc with Sarah Reisman at Caltech, which I believe will further cement the value-added nature of his contributions to synthetic organic chemistry and catalysis. It's been a great pleasure to have David in our group, and we will miss his scientific contributions and his leadership. Congratulations, David.
My favorite thing about Scripps, without a doubt, is absolutely the people. The people that comprise this institute are absolutely amazing individuals who are not only incredibly intelligent, they are also incredibly kind. They really instill each other to become the best scientists that they can become, and also just truly aspire to be interdisciplinary in their work. And for that, I'll never forget that aspect of my experience here at Scripps. For my next step, I will be joining the Reisman Lab at the California Institute of Technology to do my postdoc in synthetic chemistry. And after that postdoctoral experience, I will pursue a career as a process chemist within the pharmaceutical industry.
I'm Dale Boger from the Department of Chemistry on the La Jolla campus, and I want to congratulate Vyom Shukla. Vyom received his undergraduate degree in chemistry from Reed College in 2014, where he worked with Professors Schusterman and Gibbs, developing redshifted Bo-Dippy fluorescent dyes for super-resolution microscopy. As many may recognize, Reed College has provided some of our most outstanding graduate students through the years. Consequently, we were fortunate to capture Vyom at Scripps, and following a rotation in Jeff Kelly's group, I was thrilled when he joined our group to pursue his passion in organic synthesis. It was not only the intellectual features of natural products total synthesis that Vyom loves to be involved with, but it is also the application of the work and the problem that it addresses, which was of equal importance to him. Although the overarching goal of his work, or for his work, was in place, all of the work in his dissertation addressing the development and applications of the cycloaddition cascade of oxidiazoles was both of his conception and subsequent implementation. Simply put, Vyom is a gifted scholar, a talented experimentalist, dedicated scientist, and ambitious yet gracious in everything he tackles. On top of this, Vyom was an indispensable member of our group. In addition to his studies on the development of the cycloaddition cascade of oxidiazoles and their applications, he served as my and Ryan Shenvy's teaching assistant for modern organic synthesis for 2 consecutive years, and he served as our group manager for more than 3 years, overseeing our group, its personnel, and instrumentation, and being a mentor to all new group members. It is an unrelenting responsibility that he handled with grace and charm. He was a treasure for me and all within Scripps, within the group, and really throughout all of Scripps to work with. Vyom joined Promega Biosciences in their manufacturing group as a process design scientist, upon completing his work at Scripps. Congratulations, Vyom.
My time at Scripps taught me a lot about not only chemistry, but also about leadership, about conducting oneself in a group full of diversity, a diversity of backgrounds and also ideas, and about myself. And I credit Scripps with a large part of my personal growth over the past several years. I'd like to thank Professor Dale Boger for taking me into his lab and teaching me so much over the years, as well as all the professors I had the pleasures of interacting with during my time at Scripps, particularly to my committee members, Drs. Shenvy, Kelly, and Engel. I'd also like to thank all the members of the Boger Lab, past and present, for being a constant source of companionship over my grad school career, and all the friends I made at Scripps for keeping me sane over those long years. Finally, my eternal gratitude to my family and Whitney for sticking by me through everything without a single word of reproach towards my distant ways. I can't thank you enough for everything.
I am Ben Cravatt from the Department of Chemistry on the La Jolla campus, and it is my pleasure to congratulate Taka Aki-Ichu, who performed his thesis work in our lab. Taka was born and raised in the southern part of Japan and grew up in a traditional Buddhist monk family and lived in a temple. At 16, Taka aspired to study English and see the world outside Japan. So he moved to Victoria, British Columbia for high school. Following graduation, he studied at the University of Victoria, where he majored in statistics and minored in biochemistry. While working at the University of Victoria Genome Proteomics Center, Taka engaged in metabolomics research to identify biomarkers in tadpoles undergoing metamorphosis. Here at Scripps, Taka continued to build on his interests in metabolomics and bioinformatics, in this case focusing on studying the biochemical basis for a rare neurological disease termed FARC. Taka's work has greatly enriched our understanding of the key role that bioactive lipids play in communicating crosstalk between the immune and nervous systems, and how this crosstalk ultimately regulates brain development and health. His research findings reflect an innovative integration of chemical and biological approaches and have important basic and translational implications that include potential approaches to treat FARC and other neuroimmunological disorders. On a personal note, Taka has been a joy to have in the lab. His intellect, enthusiasm, and collaborative spirit have elevated all of those around him. This creativity and dedication that Taka brought to our lab will now be applied to create innovative medicines in his new position as a scientist at biotechnology startup in Boston, Cogent Therapeutics. Congratulations again, Taka, on this wonderful achievement.
My favorite aspect of Scripps' experience was being a member of this amazing community with friendly, brilliant scientists. Everyone from students to faculty to people in the grad office was supportive, and I could really feel that they wanted me to succeed. My advisor Ben was always supportive, and I received invaluable training from Ben and the lab. And of course, I couldn't have completed my Ph.D. without the support from my friends and family.
Hi, I'm Matt Disney. I'm a professor on the Florida campus of Scripps Research in the chemistry department. It's a pleasure for me today to talk about Matt Costellas. So Matt is a graduate student in the lab, and I think as of now has the record for publications. So Matt has been an author on 12 publications in the lab that have really, I think, highlighted his skills and talent for doing science. So Matt was able to do some very hard things that range from chemical synthesis all the way to computational chemistry. And so one of the things that I'll always remember about Matt is that at Scripps, many times you have challenging projects and you tend to initially give them to postdocs. And so Matt had worked on a project that I had given about 2 or 3 postdocs early on, and none of them had made much progress. And so I thought, hey, well, if we want to progress it, then I should just give it to Matt Costellas. And so Matt, I think, is a really remarkable scientist and has taken us in directions that we wouldn't have been able to go to otherwise. And so I'm very appreciative of having him in the lab, and I'm really excited to know what he's going to do at present and what he's going to do in the future to really change the course of science. He's now a scientist at Dicerna, and I'm sure them, like me, have been really lucky to have him working with them and also have him in their life. So thanks, Matt, and congratulations.
In a time where scientific literacy is so desperately needed, I'm happy to say I completed a rigorous scientific education at Scripps Research in Jupiter, Florida. Once you finish your undergrad research experience and go on interviews for graduate school, you feel a sense of hubris in the sort of knowledge that you have. Upon starting your graduate school experience, you immediately realize you know much less than you originally thought. However, upon completion of your PhD, you can now acknowledge how much there really is to learn. Additionally, you come away more humble, open-minded, and with the ability to truly think critically on how to solve your problems. Now, all of my achievements are thanks to many other people. My mentor, Matt Disney, gave me the chance to work in his lab, as well as provided continuous support and motivation. Scientists I've met in the Disney lab and beyond, we've all gotten smarter together, as well as fatter from all the birthday cakes that we've shared. And most of all, I'm absolutely grateful to my family and friends who have stuck with me throughout this entire process and the rest of life. Thank you very much.
Hello, everyone. My name is Kiri Engel, and I am an assistant professor in the chemistry department on the La Jolla campus of Scripps Research. It is my distinct pleasure to introduce 2 outstanding graduates from my research laboratory. First, I'd like to introduce Joseph Derosa. Joe grew up in New York City and began his career in science actually as an English major in the Macaulay Honors Program of the City College of New York. For a class assignment, he interviewed a young chemistry professor named Mark Biscoe, who managed to spark his passion for chemistry research and convinced him to switch his major to chemistry and join his research laboratory. 2 publications and an NSF graduate research fellowship later, Joe found himself bound for graduate school, and I was able to convince him to spurn offers from several other top programs in the country to join my then nascent research laboratory at Scripps Research. Joe truly lives, breathes, and sleeps organometallic chemistry, and as a testament to this, in his first year of graduate school, he had a custom plaque made for his apartment that reads, La Casa Organometallica. Exhibiting the highest levels of creativity and independent thought, Joe invented a method to improve upon a well-established Nobel Prize-winning class of reactions called cross-couplings, which typically stitch together two reactive units. Joe was able to introduce a third reaction component, namely an alkene, as a conjunctive linchpin, greatly streamlining access to valuable functionalized building blocks. Joe discovered that an inexpensive, earth-abundant nickel catalyst was uniquely effective for these types of reactions. He became a founding member of Team Nickel within my research group and wrote a successful National Science Foundation grant that will continue to fund our efforts on this topic well into the future. Joe's stellar contributions to research and to the graduate program were recognized with a 2018-2019 Turner B. and Leslie Shelton Award for Excellence in Chemistry Graduate Studies. After graduating in just over 4 years with 13 publications to his name and several more in the pipeline, in top venues, including the Journal of the American Chemical Society, Engavante Chemie, Joe moved up the I-5 to Caltech, where he is now an Arnold O. Beckman postdoctoral fellow in the Inorganic Chemistry Research Group of Professor Jonas Peters. Joe, congratulations on your accomplishments. I hope that you've learned half as much from me as I have from you over the past few years. I'm very proud to be your mentor, and I'm looking forward to watching your magical career unfold.
Graduate studies is an extremely challenging yet extremely rewarding process. And much of the success that I've been extremely fortunate to encounter throughout my PhD would really be impossible without my advisor, Kiri, and the support of my family and friends. So first off, I'd like to just thank Kiri for really believing in me at times where I honestly didn't believe in myself, and challenging me to push myself and unlock my potential as he saw it. That's something that has been critical to my daily thinking and the future of my career for sure. On top of that, my family back in New York for being so supportive despite having me go so far from them for an extended period of time, and especially the friends that I had made in New York City growing up and the ones that I've met at Scripps that will continue to be lifelong friends.
Next, I'd like to introduce Zhen Liu. As an undergraduate, Zhen Liu attended Peking University, where he quickly emerged as a prodigious researcher, co-authoring a remarkable 12 publications in the laboratory of Professor Jianbo Wang, including 2 first author contributions. After receiving his bachelor's in 2015, we were very fortunate indeed to recruit him to join the graduate program at Scripps Research. And I was especially fortunate to have him join my very first class of PhD students. Now, 12 publications as an undergraduate is really an unparalleled record. So when I began mentoring this organic chemistry prodigy, I was rather worried that I would hold him back. And after 4 years, Zhen proved that he was so talented that even a brand new professor like myself with no experience could not derail him. In my group, Zhen developed a remarkably general reaction platform to convert unactivated olefins, a class of chemical feedstocks, into 1,2-difunctionalized products. These are common structural motifs found in human therapeutics. And he found that these amazing transformations could take place using a simple, commercially available, and convenient palladium catalyst. His research culminated in 13 publications in leading journals, such as the Journal of the American Chemical Society, and he was deservingly recognized with local, national, and international awards for research, including the prestigious 2017-2018 Henry and Rose Starr Scholarship in Chemistry from Scripps Research, a 2018 Bristol-Myers Squibb Graduate Fellowship, and an IUPAC Solvay International Award for Young Chemists Honorable Mention recognition earlier this year. After receiving his PhD in just 4 years, Zhen moved up north to Caltech, where he is now pursuing postdoctoral research in the area of biocatalysis with Nobel laureate Professor Francis Arnold. Zhen, congratulations on your accomplishments. Thank you for all of your irreplaceable contributions to my lab, and best of luck in your future career. I know that you'll continue to make me very proud.
I really enjoy my study at Scripps, where I learned to become more and more passionate about science, being more creative, keep working hard and never give up. I really want to thank my supervisor, Kiri Ingo, for his help over these years. and he has been super nice to me and helped me in many, many ways. And also, I would like to thank my friends and the coworkers at Scripps, especially the 6 undergraduate students who worked with me on several projects. Without their help, I couldn't finish any of these projects. Finally, I want to thank my parents and my girlfriend. Without their support, I wouldn't be able to go through the hard times during my PhD. So thank you all.
I'm Olivia George. I am an associate professor in the Department of Psychiatry in the School of Medicine at the University of California, San Diego. Dr. Sierra Simpson investigated the role that bacteria in your guts play in the pharmacological effects of drugs of abuse, such as alcohol and opioids. She not only found that taking drugs can produce this adverse effect on the microbiome, but also that changes in the microbiome may affect the way drug acts on the brain. She identified specific bacteria in the guts and related metabolites in the blood that appears to contribute to the vulnerability to drug addiction. Her contribution to science has been outstanding, and it generated such a tremendous enthusiasm in the field and in the industry that she's spinning off a company that will use her discoveries to improve the diagnosis and treatment of drug addiction. Sierra has been an outstanding graduate student and has received many awards, including a Bagel Fellowship for her research. I am very proud of her achievements, and I am looking forward to continuing this collaboration with such an outstanding colleague. Join me in congratulating Dr. Simpson.
I think one of the most rewarding things about Scripps is always having a problem and then getting to solve it. Scripps taught us how to do that in a very untraditional and creative way. I'd also like to thank Dr. Olivia George, my mentor, for fostering my creativity and supporting all of my crazy ideas. He taught me so much about addiction neuroscience, which was a new field to me. I'd also like to thank my committee for having open doors and email boxes whenever I needed some help, as well as the graduate office for all of their hard work. I'd also like to thank Dr. Lisa Stowers, who opened her lab and her office to me throughout the entirety of my graduate experience. It meant a lot to me to have someone there to talk to when I really needed it. I can't forget my loved ones, my mother Sherry and my partner David, who flew back and forth 72 times in the entirety of grad school. Without his support, I couldn't have done this. Thank you.
First and foremost, I'd like to offer my congratulations to Gabriel Brighty on this important occasion of earning his PhD. I'm Jeff Kelly in the Department of Chemistry. Gabe joined us at the Scripps Research Institute graduate program from the University of Washington in Seattle, where he earned a BS degree in biochemistry and published a very nice paper as an undergraduate under the mentorship of Dustin Malley. I was indeed fortunate that Gabe decided to join my laboratory, wherein he probed the hypothesis that we could start with a weak but activatable electrophile in the context of an organic structure of intermediate complexity, and using affinity chromatography mass spectrometry, identify the set of proteins that are uniquely capable of reacting with the chosen small molecule. We call this strategy inverse drug discovery, as it's effectively opposite to the strategy that's often used for drug discovery, wherein approximately 1 million small molecules are screened against a protein target or a cell looking for the desired outcome. So Gabe, first working with David Mortensen, published a paper on this concept using sulfur-6 exchange electrophiles discovered by the Sharpless lab, paper that was published in the Journal of American Chemical Society. It's been cited nearly 100 times in only 2 years. Gabe went on to show the generality of this strategy with another novel but weak activatable electrophile, sulfur amide itoyl fluorides, discovered also by the Sharpless lab in a paper that's in Press and Nature Chemistry. Gabe also contributed to a paper that targets intracellular lipid binding proteins using arylfluorosulfates in collaboration with the Sharpless lab. And lastly, Gabe contributed as an author to a very nice Journal of Clinical Investigation paper examining the fraction of individuals that respond to the neurodegeneration correcting drug tafamidis that my lab had the privilege of discovering. He wanted to see if metabolism of the drug influenced responder status. What was notable about that paper is that it dramatically affected Gabe's personal life as he ended up dating the lovely Cecilia Monterio, who is the first author on that paper. So Gabe, I know your girlfriend Cecilia, your family, and your lab, including myself, are very proud of you today. You've truly earned your PhD while getting in some surfing as well. Have an awesome day. You deserve it.
You know, when I first started here at Scripps, I thought things were going to be a lot easier than they were. And as time went on, things got really challenging for me. And I really didn't think I was going to be able to finish. But fortunately, I've met some truly amazing people through my time here, people who I consider to be lifelong friends. And so, you know, I'm also incredibly grateful to Jeff for being my advisor, for his patience, his guidance, especially when things were tough. And I would like to also thank my friends and family whose love and support were really, really crucial for me to get through this process. So thank you.
So congratulations, Ryan Paxman, on this very important day as you've earned your PhD. I'm Jeff Kelly. I'm in the Department of Chemistry. Ryan came to the Scripps Research Institute graduate program from Brigham Young University, where he earned a BS degree in biochemistry and published 2 papers as an undergraduate under the mentorship of Professor Josh Price, who was a Scripps postdoctoral fellow. I was really privileged that Ryan decided to join my laboratory, wherein he's discovered a set of molecules that activate a transcriptional program that reprograms the ability of cells to maintain their proteome. Historically, scientists thought it was impossible to activate these so-called BZIP transcription factors that initiate these transcriptional programs owing to the lack of small molecule binding sites on the transcription factor. Not deterred by that challenge, Ryan partnered with the Weissman laboratory, whereby together through cell-based phenotypic screening, we discovered a few distinct molecules that selectively activated the ATF6 transcriptional program. Subsequently, Ryan and chemistry professor Evan Powers hypothesized that this molecule, so-called 147, was a prodrug that was oxidized by a P450-like enzyme to afford a very reactive electrophile that perhaps reacted with a protein that was a negative regulator of the transcription factor that we were trying to activate. So Ryan, in 2 very nice eLife papers, in collaboration with the Weissman group, provided substantial evidence for their hypothesis. And furthermore, using affinity chromatography mass spectrometry experiments, identified the reactive proteins that regulated the ATF6 transcription factor as the disulfide isomerase family. So this molecule 147 turned out to be active right out of the screening deck in mice, and in a Nature Communications paper, again in collaboration with the Weissman and Blackwood groups, we showed that 147 notably prevented heart and brain reperfusion injury through the transcriptional reprogramming of those organs. In addition, with Jonathan Lim's lab, we showed that compound 147 regulates stem cell differentiation, a paper that was published in Science Signaling. While undoubtedly Ryan has proven to be an exceptional synthetic chemist, his treasured creation, without a doubt, is his new baby girl, Claire, conceived with his lovely wife, Julie. Ryan, I know that your wife, your family, and your lab, including myself, are very proud of you as you've truly earned your PhD. Have an awesome day.
I'd say my favorite part of attending the Scripps Research Institute for my PhD was the wonderful individuals I got to interact with. Scripps has world-class scientists and postdocs. They hire the best postdocs, and I got to learn from these people each and every day and get hands-on experimental advice from them. They also have wonderful mentors who drive the lab and create unique and exciting projects to work on. That was my favorite part of attending Scripps. What I'm planning to do after I graduate is work in industry. I particularly would like to start working as a medicinal chemist or a chemical biologist on new medicines that will cure diseases, including things like the new molecular glues.
I am Cottrell from the Department of Molecular Medicine on the La Jolla campus. I want to congratulate Megan Vaughn on completing her PhD studies investigating the regulation of exercise physiology by circadian clocks in my lab. Megan has been an outstanding student. This week, her 8th manuscript was accepted for publication, and it describes the most significant portion of her research in which she discovered a direct regulation of hypoxia signaling by the circadian clock. This has major implications not only for exercise physiology, but could also impact our understanding of how circadian disruption impacts other diseases like cardiovascular disease and cancer. Not only has Megan been highly productive in the lab, she's also been a pleasure to have around. Megan has a dry sense of humor, and she's a very talented artist. Her cartoons have kept us all entertained, while her hard work has moved the scientific progress forward. A couple of years ago, Megan realized that her long-term goal is to have a career in clinical research and medicine. And so she has been accepted to multiple medical schools, and she is starting her next phase of education in medical school at the University of Arizona this summer. I'm very proud of Megan and of the work that she's done in my lab. It was a pleasure to guide her through her doctoral studies, and I look forward to seeing what she does in the next step of her journey. So congratulations to Megan Vaughn.
I'd like to thank multiple people. First of all, my PI, Katja Lamia. Second, everyone in the Lamia lab, so both members past and present, especially Alana, who's really been my partner in crime throughout all of this. I'd also like to thank everybody on my committee, which is Luke Wiseman, Enrique Saez, and Dennis Wolan, as well as my external examiner, Clara Peak. And then I'd also like to thank a couple people in the overall graduate program, such as Daphne Lurie and Paul Cognata. I really couldn't have done it without you guys.
Hello, I'm Gabe Lander from the Department of Integrative Structural and Computational Biology, and it is my great pleasure to congratulate Colby Sandate. Colby came to my lab with the intention of taking full advantage of the latest developments in a structural technique we specialize in our group called cryo-electron microscopy to study the timekeeping machinery within our cells. Essentially, these are the tiny biological timekeepers that wake you up right before your alarm clock goes off in the morning. After significant effort over the course of several years, Colby was able to shed light on how a circadian protein complex, aptly named the clock cycle complex, is regulated by numerous other protein cofactors to accomplish circadian rhythms. This work was published in eLife this past year. In addition to this work, Colby is currently wrapping up some very exciting studies on another circadian oscillator that I am confident will be published in the top-tier journal in the near future. Since these major projects weren't enough to satisfy Colby's immense intellectual appetite, he also juggled numerous other projects as a graduate student, one of which led to a publication in Nature Structural Molecular Biology, as well as a significant contribution to a review published in Nature Reviews Molecular Cell Biology. Colby was awarded a fellowship from the National Science Foundation, a Carl Storm Travel Award from the Gordon Research Conferences, and he's won numerous poster awards and spoken at international conferences. In addition to being an outstanding and driven scientist, Colby is also an incredible humanitarian, always striving to improve things for his fellow graduate students. He's served on the Graduate Student Council since his first year and eventually served as president. So it seems that even if science doesn't work out for him, I guess he could always go into politics, but let's all hope it doesn't come to that. After receiving numerous postdoctoral offers from several labs in the States and Europe, Colby has decided to go to Basel, Switzerland for this fall to pursue his studies on the cellular machinery in the lab of Nico Thoma. Given his track record and unique scientific perspective, I have no doubt that he will be wildly successful there. Colby, I couldn't be more proud of you, and I'm looking forward to your discoveries along what is sure to be an exceptional and exciting career path. Congratulations.
My experience as a graduate student was one of great growth. Not only did I learn the skills of my profession, but I also made long-lasting relationships with my peers and colleagues around the world. I owe much of this to my advisor, Gabe, who initiated me into the world of cryo-EM and taught me to be a better scientist. I also, of course, want to thank my friends and family in San Diego and back at home in Northern California. Without your support, I never would have made it this far. Thank you.
Now I have the honor of congratulating another exceptional young scientist, Mengyu Wu. Mengyu is the type of researcher that will never let anything come between her and her work, with the exception perhaps of a very cute cat video. She possesses a drive that I've never before seen in any graduate student. In collaboration with Suk-Young Lee's group at Duke University, Mengyu worked on a variety of biomedically important ion channels. Some of these are the channels that are responsible for our ability to sense heat, cold, or pain, and her contributions to this field established the architecture for therapeutic targeting of these channels to treat a wide range of diseases. While she was working on these channels, Mengyu also pioneered the use of cheaper, lower-end microscopes to obtain structures of the same resolution and quality as those obtained using microscopes that cost millions of dollars more. She simultaneously demonstrated that these microscopes could be used to examine specimens that were much, much smaller than ever thought possible with this technique. The impact of Mengyu's studies were immediately recognized as a game changer on a global scale. Her work has inspired dozens of universities, research institutions, and pharmaceutical companies in countries around the world to invest in these technologies. And thanks to her revolutionary studies, biology labs everywhere now consider routine what had previously thought to be impossible. Suffice it to say, Mengyu's time in the group was prolific. She has 8 publications with more in the pipeline, including 2 published in the journal Science and other various Nature journals. She's a recipient of the National Science Foundation, a fellowship from the National Science Foundation. She's received a Student Research Achievement Award from the Biophysical Society, numerous poster prizes at international conferences, and her contributions to the field of electron microscopy were acknowledged by a prestigious award from the Microscopy Society of America. Mengyu is currently being recruited for her unique skill set by numerous groups in academia and industry, and I'm sure that her innovative, incisive, and incredibly witty personality will positively impact the group that's lucky enough to convince her to join. Mengyu, it was an absolute honor to work with you these past few years. I wish you all the best and much, much more. Congratulations.
So when I first decided to come to Scripps, I was really excited, but also really nervous because it meant moving across the country away from friends and family. But in retrospect, I think that this is the best decision that I've ever made because I have grown so much in the past couple of years, and I've also learned a lot, particularly in my field, which is cryo-EM. But unlike cryo-EM, my grad experiences didn't happen in a vacuum, and there are a lot of people that I have to thank for getting me to this point. First and foremost, thank you to my PI, Gabe Lander. You've been an excellent mentor and a really big source of support, and I really appreciate all the opportunities you've given me. I'd also like to thank all the past and present members of the Lander Lab for being a really great group to work with, as well as my committee and Bill Anderson and JC Ducombe. And finally, I'd like to thank all my friends and family for just believing in me, and I couldn't have done any of this without you.
I'm Jean Loring from the Department of Molecular Medicine on the La Jolla campus. I want to congratulate Yao Zhang, known by her lab colleagues as AZ, for earning her PhD. I joined my lab in 2014 after having started her PhD studying crystallography. She wanted to join my lab to work on a project in regenerative medicine studying human disease. She had a steep learning curve moving from the structure of molecules to the challenges of working with human pluripotent stem cells. But I soon learned that I is happiest when she is faced with daunting problems with steep learning curves. And over the years, I've seen her conquer every challenge she's encountered. She thrives on challenges. Her thesis work was a good example. She decided to use human-induced pluripotent stem cells to understand the developmental neural defect that is caused by Fragile X syndrome, the most common inherited form of intellectual disability. She had no fear of technology and incorporated every tool available from electrophysiological analysis of neurons to single-cell RNA sequencing. And the results of her work will fundamentally change our understanding of neurodevelopmental disorders. Ai is completely dedicated to her science and to learning. We all have whiteboards in our labs and offices to outline our ideas and our experiments, but Ai is the only person I know who has a whiteboard in her home. One of the things that I like most about Ai is that she challenges me. One of Arnold Beckman's tips for a successful life was hire the best people, then get out of their way. I didn't need to teach Ai very much. I just got out of her way. I've learned more from her than she has learned from me. Congratulations, Ai. May you never stop learning and teaching. You have tremendous gifts, and there are great and lasting discoveries in your future.
I'm the first PhD graduate in my family, and I'd like to thank many people who made it possible for me. Firstly, my deepest gratitude goes to my advisor, Dr. Jin Loring. She always believed in me. Her guidance, encouragement, and support helped shape me who I am today. I also owe a great deal of debt to my mentors, Dr. Yanling Wan and Dr. Michael Boland, who have always been patient and helpful whenever I have questions. Yanling has not only been a great mentor, but also a very dear friend. I'm thankful for Dr. Adam Padaputian, who gave me great constructive advice over the years. I could not have finished my thesis work without the help of many student interns. Shout out to Mr. Josh Davis, Lee Rao, and Luke James. I'm also very lucky to be in a supportive environment like the Loring Lab, constantly getting help from Ms. Ha Tran, Candice Lynch, Aditi Shankar, Mr. Jason Ling, and Dr. Andreas Brett Liu. Above all, I owe all my achievements to my family, especially my parents and grandparents. The love and the sacrifice they made for me always motivate me to be the best version of me.
I'm Professor Ian Mcrae from the Department of Integrative Structural and Computational Biology on the La Jolla campus. And today I want to introduce Dr. Yao Xiao and congratulate her on completion of her PhD. So Yao grew up in the city of Wuhan in China, where she still has many friends and family members. She started her career in science when she moved to Peking University for undergraduate studies and research. Now, Yao distinguished herself as an undergraduate capable of advanced research by studying plants in the laboratories of Hongwei Gao at Peking University and Sumei Chen at UC Riverside. Yao then entered the Scripps graduate program, and I was fortunate enough to recruit her into my laboratory. It has been my privilege and pleasure to work with Yao over these last 5 years. The really great thing about working with Yao is that she takes science and scientific discovery very seriously. Yao pushes herself in her research, and she approaches it with a laser focus and intensity. She also never shies away from the chance to explore something new. This has allowed Yao to dive deeply into a number of projects and push the research in my lab into new and totally unanticipated areas. During her time in the lab, Yao has carried out a rigorous biochemical analyses into how small RNAs control the expression of genes in both animals and in plants. Most impressively, her studies have largely been directed by her own innate drive for discovery and innovation. I want to congratulate Yao again on completing a fantastic PhD thesis. I am proud of her work, and very excited for her future.
Hi, I'm Yao. I'm a 50th-year grad student here at Scripps with major in molecular biology. For my graduate study, I'd like to thank my supervisor, Dr. Ian Mcrae, for providing all kinds of insightful advice during the past 5 years. Also, I'd like to thank my family and friends for consistently supporting me under any circumstance. Without your help and support, I wouldn't have gone so far to earn my doctorate degree here.
Welcome, everybody, from my home base in La Jolla, California. I guess this is the new normal for all of us. Please excuse me for being a little awkward, but I will try and do justice to Chad Dufault, who we are here to congratulate on conferring his PhD degree. I'm in the California campus in the Department of Immunology and Microbiology. Chad's PhD work was exceptional. I find discoveries in science can happen rapidly and in great flash and flare, but it's actually the follow-through requires a deep dive into today's really understanding of modern biology. Now, Chad understands this principle to his core. His PhD work started with a fantastic and robust discovery of how anti-PD-1, one of our famous checkpoint inhibitors used in cancer therapy, actually impacts normal B-cell biology. This discovery required uncovering the focus of activity and looking at how this molecule impacted the evolution of B-cell immunity. This has changed our conceptual framework. The way we understand how the immune system is organized has now shifted. This work, again, is a testament to Chad's drive and insight and innovative capacity. And I really can't thank Chad enough for his impact on not only this research work itself, the follow through and changing our conceptual framework, but I think he acted as a nucleating force within the lab and within our community. So Chad was not only a force in exceptional scientific innovation, but has always been highly respected by his peers in the community, whether it's within the lab or within the department as a whole or the institution at large. Not only talking science, and certainly he'll talk your ears off on B-cell immunology, but cooking and cocktails, I think Chad is the finest resource you would find. At the end of his defense, Chad decided to actually go across the road and join Fate Therapeutics, working on advanced cellular therapies. So I know Chad will be a contributor, a major contributor to research efforts. He has the innovative drive and the sense of wonder associated with research to be a successful PhD, and I think a proud member of our community. And I look forward to seeing his research endeavors unfold over the years. So congratulations, Chad, to a well-earned PhD, and certainly our pride and joy. Thank you.
The process of getting my PhD was an extremely trying, challenging, but ultimately exciting adventure. As such, this was a stage of incredible personal growth for me. However, it was far from a solitary journey. I was lucky to have had such strong support both inside and outside of my lab during my time at Scripps. I would like to thank Michael and Louise for cultivating in me a strong attention for detail, excitement for forward-looking big ideas, and jump-starting my journey to define myself as a scientist. Thank you to my colleagues who never ran out of questions during lab meetings and made the lab such a dynamic place to be. It's hard to imagine the PhD without you guys. Thank you to my always supportive family and friends, and for coming all the way to San Diego to attend my defense. It really meant a lot. Finally, thank you, Anastasia, for being there every step of the way.
Hi, my name is Brian Pagel. I'm in the Department of Chemistry on the Jupiter campus. I relocated my laboratory here to the University of California, Irvine in 2019. I'm here to introduce to you today my graduate student, Dr. Wesley Cochran. Wes graduated from the University of California, Santa Barbara in 2013 with a degree in chemistry and biochemistry. There, his research interests included fly genetics and courtship behavior, which of course made him a perfect fit for my laboratory. He had, at the time, an interest in origins of life chemistry and enzymology, which is what drew him to me. And when he first joined my lab, Wes worked arduously developing a very complex microfluidic circuit for measuring membrane bilayer permeation. And we were really interested in this approach to develop high sensitivity permeation measurements to determine whether chiral bilayers exhibit an antioselective permeation. And we were pursuing this as a novel mechanism for explaining how symmetry might have been broken at the chemical origins of life. That work is finally coming to fruition in collaboration with Dr. Juan Hu, a postdoctoral fellow in my laboratory, and Professor Donna Blackman there on the La Jolla campus at Scripps Research, California. In the intervening time, Wes has wasted absolutely no time being what I've described as a Category 5 hurricane of science, involving himself in the main thrust of my laboratory, which has been in the development of activity-based DNA-encoded library screening technology as a new platform for drug discovery. And Wes's contributions here have been absolutely extraordinary. He was the first to show that we could put together all of the various microfluidic components that my laboratory developed to screen a real drug-like DNA-encoded library developed in collaboration with Roche Pharmaceutical Research Early Development Group in Basel, Switzerland, to discover novel scaffolds that are active against the clinically relevant phosphodiesterase target autotaxin. He is also wrapping up what will be another amazing achievement in the field, which is the development of phenotypic DNA-encoded library screening technology with a specific eye toward the discovery of novel antimicrobially active compounds. So that work is forthcoming as well. In recognition of his achievements in my laboratory, he's received the Ferris Foundation Fellowship. Wes, I cannot say enough about you. Thank you so much for being such an amazing student. I wish you all the best in your postdoctoral fellowship studies there with Jerry Joyce at the Salk Institute. I know that our continued collaboration is going to be nothing but fruitful, and I wish you all the best. Good luck. It's my pleasure to introduce to you today Dr. York Zang. York graduated from Johns Hopkins University with a degree in chemical and biomolecular engineering in 2009. He joined Scripps initially in 2012 in the groups of Wolan and Srinivasan, but then shortly thereafter transitioned over to chemical biology with Romsburg. And it was there where York made amazing contributions to science, working with Romsburg both to describe the first semi-synthetic organism that incorporates unnatural base pairs into its DNA and faithfully replicates them, but then later in a landmark paper describing the full utilization of that artificial genetic code to encode unnatural amino acid containing proteins in that semi-synthetic organism. This is a huge achievement for science, and for it, York has received both an Ellen Browning Scripps Foundation Fellowship from Scripps Research, and he's also received the highly prestigious F31 Ruth L. Kirchstein National Research Service Award from the National Institutes of Health. I came to know York as a TA in my class, Effective Scientific Communication, and York took the very first offering of that course and was an amazing student. He has a natural gift for communicating science, and he then TA'd for me subsequently, passing on his skills to future generations of scientists, offering them his insightful critique into their presentation skills, as well as all of the papers that we discussed as a class across the full gamut of biomedical research, from organismal biology to chemical synthesis. York, in his final years, transitioned over to the startup company Synthorx, where he is working as a scientist to translate his pioneering efforts in semi-synthetic organism engineering into commercial products. York, it's been an absolute honor to know you as a mentor over all of these years. I hope that you'll keep in touch, and good luck at Synthorx.
Hi there, I'm Christoph Rader from the Jupiter, Florida campus of Scripps Research, and I have the pleasure of introducing Emil Fischer. Emil hails from Denmark and received his BS and MS degrees in biotechnology from the University of Copenhagen. He carried out his thesis project in Floyd Romsburg's lab, and I was fortunate to witness his incredible intellectual and technical skills, first as a member of his advisory committee, and then as an administrative advisor in his final year. Believe it or not, Emil successfully defended his thesis with flying colors after a little more than 3.5 years in our program. Can one be productive in this short time? Emil sure was, with a brilliant first author paper in Nature Chemical Biology and co-authorships in one Transformative Nature and 3 JACS papers. All of this, of course, revolved around the development of semi-synthetic organisms, specifically the incorporation of unnatural base pairs into DNA, their transcription to messenger RNA, and translation to proteins with non-canonical amino acids. In fact, Emil has made history by generating the first semi-synthetic organism that uses 67 independent codons. Thus, in a sense, Emil has not only earned his degree, but in doing so also attained divine status, somewhat of a demigod with a PhD. That said, Emil is very grounded and longed for a return to his native Denmark. He's already back in Copenhagen, where he is currently enjoying the long summer days and preparing for a career in the industry. I hear that Novo Nordisk is interested in hiring him. And what can I say? This should be a no-brainer. Congratulations, Emil. Well done.
Hi, I'm Gavin Rumbaugh, and I'm a professor in the neuroscience department on the Jupiter campus. And I'm here to offer my congratulations to Murat Kılınç for successfully completing his PhD in my laboratory. Murat was a fantastic addition to my lab. He came from a fantastic background where he did some work at the Rockefeller, and when he came in to my laboratory, he successfully competed for an Autism Speaks Fellowship, which are very, very competitive. And with that fellowship, he went on to publish several really nice papers in my lab, and for that we are, I'll always be thankful for having Murat in my laboratory. Murat was a man of many talents. He's kind of a big guy, but he was pretty mobile. And the lab for a while did a lot of paintball here in South Florida. And with Murat's size and mobility, he pretty much acted as like a mercenary. He, I think, was pretty much always on the winning team, and in fact caught me a few times in the side of the helmet. So that was something I really, really remember about Murat. He's off to bigger and better things. He's already started his postdoc at the Gladstone Institute in the Bay Area, where he'll be studying the molecular mechanisms of neurodegeneration. So again, congrats, Murat.
I am Erica Ullman Sapphire, professor in the Department of Immunology on the La Jolla campus. And I'm here today to congratulate Liam Barry King on the successful defense of his doctoral dissertation. Liam's PhD is a first for Scripps Research. Liam's research spanned 3 diseases, 4 countries, 3 continents. Liam worked on Lassa virus in Sierra Leone. He worked on Ebola virus in the Democratic Republic of the Congo. He worked on malaria in Brazil, and he did high-resolution structural biology in my laboratory in La Jolla. Now, we all know that Scripps does high-caliber biomedical research, with a typical focus in the biomedical pathway of disease and discovering a new drug that will arrest that pathway. But Liam's PhD goes broader than that. Liam is a future physician scientist who earned his PhD here on the way to a dual MD PhD degree. He's interested in not just knowing the drug to be delivered, but understanding the clinical care needed to receive that patient and deliver that drug. For resource-poor settings, there are very hard lessons to be learned. It doesn't matter if you have a first-in-class antibody and the best in the world if the patients all die on the way to the clinic to receive it. For Liam's MD PhD, he has indeed focused on developing better methods for patient care, better access, and delivering those medicines needed. And he's focused his work where his talents are needed the most, emerging infectious diseases in impoverished people. Liam is a future infectious disease physician. Now, more than ever, researchers like Liam are needed in the world. My laboratory and our institute are both the better for having enjoyed the power of his research and the pleasure of his company for the last several years. Liam, congratulations to you, your family, and your partner, who are all celebrating with you today.
I'm really honored to be able to introduce to you 2 students. One of my graduate student, Anzi Yao, and one of Floyd Romsberg's students, Vivian Diane. Both have spent the time here at Scripps working on a very interesting problem, which is the expansion of the genetic code, which has been conserved for billions of years. Anzi came to Scripps from Peking University, where she worked in developmental biology. At Scripps, she decided to join my lab in our effort in synthetic biology, focused, as I said, on expanding the genetic code. Anzi worked on a number of problems. She's been able to add new non-canonical amino acids to the genetic code of bacteria that allow us to make cell proliferation dependent on the presence of a small non-canonical unnatural amino acid building block. We're using that as a way to develop conditional live vaccines, which could have a large impact, especially in today's public health climate. Hanzi also developed new sensors to sense various protein activities in cells using, again, genetically encoded non-canonical amino acids. And finally, she went in the other direction and asked whether one could actually begin to remove one of the natural 20 amino acids from the code, and made tremendous progress in doing that with the genetic code of bacteriophage. Anzi, please step forward.
As a grad student from Scripps, I was so grateful that I worked with one of the best advisors. Pete is a great scientist and also knows what has real impact. He constantly asked me to take on new challenges, and I was able to learn a lot from all these hard tasks, including how to do science and to be persistent. I was also very lucky to be surrounded by very supportive and inspiring lab mates and colleagues from Scripps. Finally, I want to have a special thank you to my family, friends, especially my parents and my fiancé Li Yang. I wouldn't be able to finish my PhD without your unwavering support and encouragement.
Next, I would like to introduce Vivian Diane. Vivian came to Scripps from Berkeley, where she carried out undergraduate research with Carolyn Bertozzi. At Scripps, she worked in the lab of my colleague Floyd Romsberg on another aspect of the genetic code, and that is not only adding additional unnatural amino acids to the code, but additional building blocks for DNA. So Vivian was able to create new hydrophobic base pairs, genetically encode those in DNA. Those were then transcribed to RNA, and then they were translated to proteins. In doing so, she created the ability to make a whole new set of codon-anticodon pairs that allow you to expand the genetic code not only by 1, 2, or 3 amino acids, but potentially many, many more. Vivian, please step forward.
Graduate school has been one of the most challenging, exciting, and rewarding things that I've ever done. And I'd like to personally thank my advisors, Pete Schultz and Floyd Romsberg, for the opportunity to do my graduate research. In addition, I'd like to thank my friends, family, and my boyfriend Kevin, as well as our cat and dog, Loki and Lady, for their love and support through the last 5 years.
I am Professor Barry Sharpless, bringing greetings from La Jolla. The year I became a chemistry professor, 2 graduate students joined my group. Now it's precisely a half-century later, and my final 2 students are receiving their PhDs. Gencheng Li just became a research investigator at Insight Pharmaceutical in Wilmington. He says the Delaware woodlands remind him of rural South China, where his remarkable journey began. Boundless enthusiastic energy and drive, and a family culture of humility, helping others and being respectful. These helped him overcome crushing obstacles. Gensheng's family couldn't afford their village school, so he attended one 3 mountains away over, where his grandmother was born, and at the age of 8, he just started the twice daily hour-and-a-half walk. His drive to have a better life was made fiercer by the stigma of poverty. When Genchen joined my group at Scripps, our recent discovery of suffix reactions based on sulfur fluoride gases was transforming click chemistry, a process-based method for finding function I created in the 90s. He started out making compounds from SOF4, hundreds of them, for the group's use and for suffix libraries we shared with many collaborators in biology. At the bench, Genchen was a phenomenon with famously great hands, the chemistry equivalent of an Olympic gold-winning athlete. He helped develop the new gases for suffix, a feat that needed not only flawless technique, but also fearlessness. After 3 intense years immersed in mapping out the virgin chemistry of these rare sulfur oxyfluoride gases, I asked him to create unprecedented chemical libraries for Mike Petrossi's and Scripps Caliber's multi-drug resistant tuberculosis challenge. There are about 500,000 new cases a year of multi-drug resistant tuberculosis. In his search for a cure, Genshin made dozens of drug cores and directed the SAR probing towards the best leads. Many hundreds of compounds were made and then screened by Caliber. A uniquely potent covalent drug candidate for MDR-TB emerged and is now moving forward quickly. Genshin is first author on several of his papers. However, much of his research remains unpublished because he selflessly chooses to push ever onward instead of writing papers. Hearty congratulations and best wishes to Dr. Zhang Li, an idealist and highly gifted synthetic chemist with a bright future in pharmaceutical research.
It has been a wonderful journey in the past 5 years at Scripps. I have so many good memories. I really enjoyed the Ocean View office and the spectacular sunset every day. Of course, the most important part is those great people I have met here. Barry is such a great mentor, and he's always willing to spend hours and hours with me talking about chemistry, answer my stupid questions, and show me the importance of a strong influence in scientific work. And whenever I have some trouble, I feel like I can always turn to him for help. So thank you, Barry. Also, I want to thank my friends here, and our friendship really helped me a lot, go through those ups and downs in the years. And finally, I want to thank my families, my parents and my sisters, and for their love and support. Without them, I can never be who I am.
30 years ago, I was a founding member of the chemistry department. This year, Jin Hain Zhang, a final Sharpless Group student, receives his Ph.D. Arriving not long after suffix chemistry's discovery, Jin Hain explored the suffix's reactivity, catalysis, and functions. His projects integrated click chemistry with polymer synthesis, radiosynthesis, imaging, and drug discovery. Hence, his best discoveries are interdisciplinary. But behind all these accomplishments stands a brilliant chemist. One of his breakthroughs underscores how suffix click chemistry, like the QAC class of click chemistry before it, represents a second, new, and near-perfect family of reactions. Both of these new reactions, QAC and suffix, were discovered in our group here at Scripps Research. The almost perfect fidelity of these two transformations becomes suddenly very important because we are finding that the high throughput synthesis of chemical libraries for direct biological screening fails badly unless the module annealing step succeeds perfectly. Jin Hain proved his method's power by overnight synthesis testing of a large suffix-made library, whereby the original potency was increased 480-fold, down to a Ki of 18 nanomolar. This was the first time suffix's full power for drug discovery became apparent. Jin Hain has shown himself to be a natural-born click chemist. The necessity of near-perfect reactions, the modular structural themes of molecular personalities, the very fast assembly of the modules, and the instant functional screen test of each crude product in its well. He feels these in his bones. Once upon returning from a long trip, Jin Hain's smug report signaled he knew he was onto something good. What he had, to my surprise, was a breakthrough from somewhere beyond my reckoning. Right away, I tried to make sure he knew how important it was. Since then, knowing that his ideas could be first-rate, his confidence has taken off with his ambitions toward riskier goals. Working with Jin Hain and seeing him mature has been a joy. He is now a postdoc with UCSF's eminent Kevon Shokat, professor and chair of cellular molecular pharmacology. After his postdoc, he aspires to be an academic, which I support enthusiastically. He leaves an enduring legacy at Scripps Research. So please join me in thanking, congratulating, and wishing Jin Hain Zhang even greater success in the future.
My PhD research has been focused on making the intermolecular connections around sulfur 6 hubs. On a personal level, I was fortunate to have made connections with a number of great people at Scripps. First and foremost, I would like to thank my supervisor, Professor Barry Sharpless. Studying chemistry with Barry has been the best experience for me at Scripps. I've learned a lot and continue to learn from him. I'm also grateful to my collaborators, Professors Peng Wu and Professor Dennis Wolan, and my friends at Scripps, especially Professor J.P. Lee, Drs. Yi Yang and Dr. Genchen Lee. I would like to share this joyful moment with my family, my parents, Mr. Zhen Jianlei, Ms. Jin Manhong, and my girlfriend, Ding Zhao, for their love and support. Last but not least, congratulations to all of my fellow students of the class of 2020, and I wish everyone good luck.
My name is Ryan Shenvy. I'm from the Department of Chemistry on the La Jolla campus, and I'm here to recognize Dr. Robert Martin Desmarais for his groundbreaking work in the synthesis and interrogation of the neuroactive plant metabolite, bilobalide. Rob, you trusted me with 4 years of your life, bestowed upon me your Midwest work ethic and your generally good-natured demeanor. You lent me innumerable Excedrin migraine tablets, and you even let me borrow your wife, Brooke, to teach my kids piano. For all these things, and for never having to hear you call me boss man again, I will be forever grateful. Rob joined my group with a degree in biochemistry and with a passion and talent for hardcore chemical synthesis. He teamed up with a postdoc and another talented graduate student, Megan Baker, to tackle a very tough, long-standing challenge to develop a short artificial route to metabolites of the ginkgo biloba tree. Rob, I hope that what you achieved and the lengths you've gone to achieve it will stick with you for the rest of your life. No challenge is too great, given enough patience, thought, and hard work. The metabolites of the ginkgo biloba tree have long been pitched with very poor evidence as cognitive enhancers or nootropics. Our lab was not drawn to these specious claims, but instead to the observation that one metabolite in particular could decrease GABAergic signaling in the brain without the common off-target effect of increased bleed time from antagonism of platelet aggregating factor. The problem, however, was that this special metabolite called bilobalide is like a fragile sculpture. It's unstable in blood and water, and it's very difficult to purify from the plant. So Rob and his teammates devised a way to make it from scratch, which ended up illuminating some very interesting chemistry and more recently some intriguing biology in rat brain tissue in the lab of our colleague, Professor Marissa Roberto. Rob is co-first author on a recent paper in the prestigious journal Nature, and co-first author on what I consider a magnum opus manuscript that combines synthesis, mechanism, informatics, and biology to describe Rob's exploration in the distant reaches of chemical space. So he can add these papers to his many successes in graduate school, including marrying his fiancée and my piano teacher, Brooke Anderson, recovering from a sliced tendon in his hand, befriending nearly every member of the Scripps community, and securing a coveted position at Vertex in San Diego. Congratulations, Rob. The best is yet to come.
All in all, I think there are a lot of ups and downs at Scripps. There are probably a few more downs than ups, but in the end, it's definitely worth it. So with that being said, I need to thank everybody that helped me get here, which is my friends and my family, and especially my mentor, Ryan Shenvy, for teaching me and getting me through all the hurdles that kind of came through the projects that we worked together.
It's an absolute pleasure to congratulate Dr. Samantha Allison Green for her incalculable contributions to my group's research program and to the chemistry community at large. Sam, you entered my lab fresh from a post-bac at NIAID with an incredible fund of knowledge in chemistry and biology and determined to soar at the interface with medicine. And instead, I sent you down to the sub-basement of transition metal catalysis. Yet, Sam, you excelled, scrabbling in the dark for anything valuable and emerging with a fistful of diamonds. So whenever you feel like you can't overcome, you can't solve a problem, know that you already did, and so you will. Sam addressed two long-standing problems in chemistry: how to achieve general branch selectivity in alkene hydroarylation, and how to interface HET catalysis with non-traditional radicalophiles. That's kind of a mouthful, so I'll break it down for the general audience. Many commercial products, things that we use every day, plastics, perfumes, flavors, medicines, come from unsaturated hydrocarbons, which are constituents of plant extracts, crude petroleum, and fats. By and large, however, these carbon chains engage with common industrial catalysts only at their terminus, and not in their middle. Sam found a general way to alter this selectivity and coerce the carbon chains to branch out, inventing not one, not two, but three distinct cross-coupling reactions. At least one of Sam's reactions has already been implemented by Merck on very large scale to access a lead compound in drug discovery, less than a year since its publication. And this was just one of 7 high-profile papers that Sam published during her time at Scripps. She laid a foundation for years' worth of work. And you probably don't know this, Sam, you helped secure 2 independent grants from the National Science Foundation. She was truly a force to be reckoned with and will sorely be missed. Last December, Sam graduated and, amid many offers, chose as her top choice a premier position with Genentech, designing the next generation of new therapeutics. Sam leaves behind a legacy of incredible science and has paved the way for so much more. Congratulations, Sam. You earned it.
Reflecting on my time at Scripps, I wanted to give a big thank you to my advisor, Ryan, who always encouraged us to think creatively and helped to shape how I approach problem solving today. Thank you to the rest of the Shenvy lab, who despite the long hours, managed to make grad school a really enjoyable place. And finally, thank you to my family and to Julian for their unconditional support throughout the years.
My name is Dr. Linda Sherman, and I am very pleased to introduce to you Marguerite Johnson, better known as Daisy. I am standing in for Daisy's mentor, Wendy Havren, a beloved colleague and friend who recently passed. As I observed as a member of Daisy's thesis committee, she's intelligent, inquisitive, enthusiastic, hardworking, and thoughtful in her approach to science. But I have asked Deborah Witherton, who is a staff member in Wendy's lab, to fill in some of the details that could only be known from the perspective of a colleague and lab member. And so I would like to read some of Debbie's comments. Daisy joined the lab from Texas A&M when she obtained her master's degree, studying inflammation and laminitis in horses. Although her love for horses continued in San Diego, riding her own horse several times a week early in the morning before coming to the lab, she quickly adapted her research to a much smaller animal model. Over the course of her time in the lab, she successfully juggled several challenging projects simultaneously and has 3 manuscripts at various stages of preparation and review. She effectively supervised undergraduate interns while still moving her own project along at a steady pace. Daisy was also an active participant in outreach activities, hosted lab tours and hands-on research experiences for visiting high school students, such as Science Saturday participants, teachers, Girl Scouts, and ARC members. Daisy was an ARC scholar from 2015 to 2019. On a more personal level, Daisy was a delight to have in the lab and always ready to lend a helping hand. She's usually the first to volunteer when something needs to be done.
I have so many people to thank for their support in the process of pursuing my PhD, especially my parents, Susan and Lee, and my husband, Al, who have all supported me so much through these ups and downs. But here, I really want to take a minute to talk about my amazing mentor, Wendy Havren, who very sadly passed away this January. Wendy was everything that I could have hoped for in a mentor. There are just so many things that you could say about Wendy, who's truly in a class of her own. But I think what I take away the most is how she always got the best out of people in her lab while remaining firmly in their corner. Sometimes things don't go well for many months at a time in graduate school, and it can be so hard to keep on giving it your all. But one of the main things that kept me going in the face of all of this failure was the way that Wendy could impart her belief in her students. I always wanted to live up to how Wendy saw and introduced me as a scientist, and I will be forever grateful to her that that was my experience of graduate school.
I'd also like to introduce to you Joseph Mcgraw, or as we know him, Mike Mcgraw. As a member of Mike's thesis committee, I am standing in for his mentor, Wendy Havron, who recently passed away. I've always found Mike to be highly intelligent, confident, and hardworking. But I was not a member of his lab, and so have asked Debbie Worthington from his lab to provide a perspective as a member of the lab and colleague, and these were her comments. Mike first joined the lab as a summer student in the SURF summer internship program in 2014, and from the very beginning showed himself to be an outstanding student who not only had excellent bench skills, but was also a quick learner who easily understood the concepts behind his project. Even as just a summer student, he was a dedicated researcher and a pleasure to have in the group. Wendy and all of the lab were thrilled when he decided to return to TSRI as a grad student, and even more pleased when he chose Wendy's lab to join for his PhD project. Mike was very quiet and a studious member of the lab. He was always very focused on his projects, keeping up with all the latest literature and finding new ways to address the problem in hand. He always stood out for his intelligence, excellent communication skills, maturity, and strong work ethic. Loves science and is very self-motivated. Although quiet, he got along extremely well with all of the colleagues, both within the lab and within the entire department, and was also equally adept and seeming very comfortable interacting with the faculty and with his clinician collaborators. Mike was an ARC scholar from 2019 to 2020.
My time at Scripps has been incredibly rewarding. I was lucky to be able to work with the late, great Wendy Havron. And Wendy helped make sure that I got the most out of my time at Scripps. And I certainly would not have made it to this point without her. I will always remember her passion for science and her incredible kindness, and I really wish she was still here with us today to celebrate. While this year has certainly been very trying, I would like to thank my thesis committee and lab mates for all of their support. I'd especially like to thank Debbie and Daisy, who have been there to help me since day one of grad school and have been instrumental in helping me cross this finish line. And then last, I'd like to thank all of my family and friends outside of Scripps for their love and support and helping me escape from lab every once in a while and stay sane.
I'm Andrew Su from the Department of Integrative Structural and Computational Biology on the La Jolla campus. I want to first congratulate Jake Bruggeman for completing his PhD degree. Jake's thesis research focused on developing a tool, which he calls Mercator, which can visualize and analyze gene expression from tens of thousands of samples from public data repositories. Jake's work revealed some fascinating insights that would only have been possible with this sort of large-scale data mining, and he's demonstrated its utility in a variety of different contexts, including cancer subtyping and stem cell differentiation. Jake came to Scripps with a very unique background, having completed undergraduate degrees in physics and computational and applied mathematics. He developed his interest in biology and bioinformatics relatively late, but he has quickly caught up with his peers. In fact, I think Jake stands out as one of the most independent graduate students I've ever supervised, leading his project from conception to design to execution since almost the day he joined the lab. I'll miss Jake's contributions to the lab and congratulate him on a very successful graduate career.
Being at Scripps has been one of the greatest experiences of my life. It's been so challenging and so rewarding, and I've learned so much being here. I'd like to thank my advisor, Andrew Su, for being such a great mentor, teaching me so much, making sure I got through this alive, and really making sure I made the transition from undergrad to graduate so easily. I'd like to thank the entire support here at Scripps, including the IT staff, graduate office, and the HPC people for keeping the compute cluster running. And I'd also like to thank my friends and family for being such a great support system and always believing in me.
Next, I would like to congratulate Toby Lee for completing his PhD degree. Toby's thesis included work on a broad range of topics, including crowdsourcing, text mining, and machine learning. He capped off his thesis work mining a data set of insurance claims data. This data set includes billions of medical and pharmacy claims, And Toby showed how this data set could be used to identify drug repurposing candidates based on statistical associations between drug prescriptions and disease occurrence. On this last project, my lab had no prior experience working with insurance claims data, but in collaboration with Nathan Weininger, Toby enthusiastically jumped into this analysis. He quickly digested a whole new set of literature and methods associated with this area, and he was able to adapt his skill set in a way that I think few other students would have been able to. Toby will shortly be starting a job as a data engineer at TetraScience, a lab automation and data integration company in Boston. Congratulations, Toby. Next, I'd like to congratulate Mike Myers for completing his PhD degree. Mike's thesis research focused on computational drug repositioning using knowledge graphs, which are network-based data structures that are gaining popularity in a wide variety of contexts. Mike developed infrastructure to efficiently create biomedical knowledge graphs and also machine learning methods to mine these knowledge graphs. Mike started at Scripps doing experimental work in metaproteomics, but he joined my lab as his interest in bioinformatics grew. Although he's left the wet bench behind, he's managed to combine a keen biological intuition with an outstanding bioinformatics skill set, which is a powerful combination. Mike has agreed to stay in my lab as a staff scientist to extend his work on drug repositioning for a new grant on Alzheimer's disease. Congratulations, Mike.
I've greatly enjoyed my time at Scripps and the freedom offered to me to explore different areas of biomedical research. Of course, I'd like to thank my advisor, Andrew Su, for the guidance and freedom he gave me over the years. And again, big thank you to everyone in the Su lab for all the help and support you gave me when learning bioinformatics methods. And thank you to my parents and to my wife, Julianne. Without your love and support, I would not have made it through this program. Again, big thank you to everyone.
Finally, I would like to congratulate Ryan Thompson for completing his PhD degree. Ryan's primary project involved the study of the genomic and epigenomic basis of T-cell activation. His analyses revealed important insights into the interplay between histone modifications and gene expression, and how those patterns differed between naive and memory T-cells. I want to acknowledge Professor Dan Solomon's instrumental role in Ryan's thesis project, who served as his mentor until 2016, when he unfortunately passed away. And also acknowledge Ryan's perseverance in completing his degree under these challenging circumstances. Finally, I'll note that Ryan not only has an outstanding ability to understand and apply complex biostatistical methods, he is really gifted at communicating the intuition behind these methods to non-statisticians. The Scripps community has really benefited from that gift, from my lab to the bioinformatics core to the graduate school through guest lectures. Earlier this year, Ryan started a position as a data science analyst at the Mount Sinai School of Medicine. Congratulations, Ryan.
My path through graduate school has been a long and winding one, and I'm grateful to all the mentors I've had through the years. Doctors Terry Gasterland, Dan Salomon, and Andrew Su, whose encouragement and support have been vital to my development into the scientist I am today. I couldn't have done this without any of them. I'm also thankful for my many collaborators in the Salomon Lab and elsewhere. Finally, I wish to thank my parents for instilling in me a passion for science and learning from an early age, and encouraging me to pursue that passion as a career as I grew up. I'm truly lucky to have such a loving and supporting family.
I'm Mark Sundrud from the Department of Immunology and Microbiology on the Jupiter campus, and I'd like to congratulate Meilan Chen as a part of the graduating class of 2020. Meilan came to my lab shortly after I started, and as an undergraduate chemistry major, had very, very little biology experience. But after 10 weeks in the lab, she was already purifying ribosomes and characterizing transcripts in T cells that were associated with those ribosome fractions. It was abundantly clear at that point that Meilan had everything that was needed to become a really creative and successful scientist. So for the last 6 years, Meilan has been drawing together seemingly disparate concepts in everything from mucosal immunology to gastrointestinal physiology and infection biology into cohesive, important new mechanisms that underlie how T cells maintain homeostasis during immune activation and inflammation. The work from Meilan has resulted in numerous important papers, as well as equally important funding for the lab. Although I'm very sad to see her go, I'm also equally excited to watch her as Mei-Lan makes a name for herself and continues to make an important impact in the biotech and pharmaceutical industry. So congratulations, Mei-Lan, well deserved.
My experience as a graduate student at Scripps was an adventurous and challenging journey. My advisor, Mark Sundred, helped me develop my scientific insight and technical knowledge to become an independent researcher, and I will be forever grateful to him. I would like to thank my mom and my mother-in-law for all the support throughout the years, and of course I'm thankful to my loving husband, Christian, and my daughter, Frances, for all the support, hope, and encouragement.
Hi, my name is Andrew Ward, and I'm a professor in the Department of Integrative Structural and Computational Biology here at Scripps on the California campus. And I'd like to start by congratulating Christopher Cottrell, who is a graduate student in my lab who recently defended his thesis. Chris is actually a local from Poway. He still lives in Poway, and he went to undergraduate at the Naval Academy. And after 4 years there, went to Johns Hopkins and got his Master of Science in Biotechnology. After that, he spent a couple years at the Fred Hutch, working in the lab of Julie Overbaugh, and really developed a passion for HIV and HIV vaccine research. He joined the graduate program, and I was fortunate enough that he wanted to join my lab and work on structure-based immunogen design for HIV. So Chris has done a remarkable job across a broad spectrum of projects, including HIV and coronaviruses. He solved some of the first structures of coronavirus spike proteins in the lab just when he was starting out. So this is all culminated in almost 40 manuscripts that will be part of his PhD thesis, and really a legacy that's going to be hard to top. He set the bar in various aspects of my lab, is a mentor to many new students, and really the go-to person for all things immunology. He's really helped transform my lab from structural biology to immunology and beyond. And so with that, I'd like to congratulate him and his family and his friends that are all joining in this celebration. And next, I'd like to introduce and congratulate Bartek Nogal. Bartek did his undergraduate and his master's at Cornell University on the East Coast. And then he had a short career in industry that spanned Merck, Genentech, Novartis, and Gilead, where he served different roles in those companies and really developed a deep understanding of process, biochemistry, and development. And he decided then that he'd like to pursue his PhD. Again, I was fortunate that he joined my lab and really had an interesting background. He came to the lab full of new insights and really took on some new endeavors in imaging and microscopy in terms of epitope mapping, antibodies on the surface of HIV envelope for the purposes of structure-based vaccine design. Bartek was a student who had an interesting schedule. He was raising 3 children at the same time as taking on, a very difficult PhD. So we didn't overlap that much because he worked nights and weekends, but I'm happy to say he did fantastic. I know he appreciates the support that the lab gave him in doing those sort of 2 jobs simultaneously. Bartek has now moved on to a small startup biotech company on precision medicine, and I'd like to congratulate him as well.
It's my pleasure to introduce and honor my graduate student, Yisong Deng. Yisong came to my lab 6 years ago, and he has done a remarkable thesis studying the assembly of the HIV particle. Yisong's project was a real tour de force that required a number of different components. First of all, he had to design mass spectrometry experiments to detect minuscule amounts of the viral protein that was being expressed and assembled inside the cell. And second of all, he had to develop analytical methods to even understand what this mass spectrometry data was telling us about the pathway of virus assembly. Yisong took this project on to investigate a major paradigm in virus assembly, which was the assembly of the HIV gag protein into a series of intermediates in virus assembly. His work basically tore the underpinnings of this work completely apart and came up with a completely new model. Essentially, the major paradigm in the field was overturned by his insightful biophysical characterization of the virus assembly process. Yisong is a delight to have in the lab. He's extremely funny. He has a dry sense of humor. He's extremely well liked. One of the most pleasurable students I've had the opportunity to interact with. And on top of that, he delivered a killer thesis, establishing a new paradigm for HIV assembly. So to his family, his parents, congratulations. Very proud of your son, and I wish him well in his future endeavors. Congratulations, Yisong.
Training under Dr. Jenny Williamson has been a blessing for me. I'm always amazed by his vast knowledge and creative ideas. I feel lucky to be able to explore different fields under his guidance. The past 6 years has been a journey. I learned many valuable lessons about science and my life, and have received countless helps from the people around me. I really wish to thank my collaborators and lab mates who teach me many things about science and research. I wish to thank my friends who spice up my life and explore San Diego with me. I also wish to thank my family who provides me unconditional love and support. I can hardly imagine being here without any one of you.
I'm Dennis Wolan. I'm in the molecular medicine department here on the La Jolla campus, and I am here to introduce Janice Zhu for her PhD in chemical biology. Janice received her Bachelor's of Science in Chemical Biology at the University of California in Berkeley in 2015. And for her final 2 years while she was at UC Berkeley, she worked with Gary Firestone, where she was looking at biochemical characterizations of indole-containing compounds and their effects on the human ubiquitin ligase NED4, and how that function and disruption of that function affects human melanoma cancer cells. She joined my lab in August 2015 when she entered TSRI, and she entered into the chemical biology program. And in 2016, Janice was a National Science Foundation Fellowship honorable mention. And in 2017, she received an International Proteolysis Society travel fellowship where she went to Banff, Canada to present her poster on some of the work that she was doing in the lab at the time. Janice is a jack of all trades, basically. Her primary focus has been on protein structure, primarily protease structures, and biochemistry. And she's been tasked with multiple projects in the lab, working primarily with crystallography, drug discovery, and mass spectrometry. She was the first person to determine the structure of a proform of the enzyme caspase 8, which is involved in apoptosis. And she did this in the presence of a small molecule inhibitor that was discovered by Carrie-Ann Bacchus, who is an assistant professor at UCLA. So in collaboration with Carrie, as well as Stefano Forli here at Scripps, we published a paper with Janice as a first author in ACS Chemical Biology. In collaboration with Anthony O'Donoghue and Vivian Hook over at UCSD, Janice determined the crystal structure of a protease we call PD dinase, which is involved in the secretion from a bacteria that we have in our human distal gut. And so completely on Janice's idea, she determined what the protease's specificity was, as well as what the actual biologically relevant substrates would be in our human distal gut. And Janice found that these were, in fact, human antimicrobial peptides that we secrete as a defense mechanism to sort of keep bacteria away from our bodies. And so this was an example of one way in which our commensal organisms probably colonize our gut, and how we can differentiate between what are commensal healthy organisms versus those that are pathogenic. And she also did some work with Enrique Sayez and Ben Kravat's lab looking at a small molecule activators of a protein called human lipol A1. And that paper was just recently published in Nature Chem Bio. And throughout all of this, Janice also volunteers at the San Diego Humane Society. Janice is going to continue working on, for the time being, finishing up her projects in the lab. And then ultimately, she would like to pursue her plans to look into product development and regulatory affairs within the biotech industry. So I would just like to thank Janice for all of her persistence, intuitiveness, and commitment to the laboratory over the last 5 years. It was just a fantastic opportunity to have Janice as a colleague over the last several years, and she's going to be severely missed. So I just want to thank Janice and her family and congratulate them on an amazing accomplishment.
I'm extremely grateful to my advisor, Dennis, for his guidance these past 5 years. His mentorship, optimism, and kind encouragement have been inspirational and crucial for my growth as a scientist. These past 5 years at Scripps have been quite a roller coaster, and I wouldn't have made it to the end without my family and friends. I'd like to thank my parents and brother for their unwavering support, and I'm greatly indebted to my parents for their hard work and sacrifices that have allowed me to complete this PhD. My food adventures with Angelo, Peter, Anna, and Gonzalo were some of the highlights of my graduate school career. And I'd like to give a big thank you to Vivian, Christine, Sabrina, Oliver, Sophia, Dorothy, and Isu for their years of friendship and fun.
I'm Peter Wright from the Department of Integrative Structural and Computational Biology on the La Jolla campus. I have great pleasure in congratulating Sergei Schnitkind on attaining the degree of Doctor of Philosophy. Sergei was born in Russia and grew up in Israel. He completed a Bachelor of Science degree in biochemistry at UCLA before joining our graduate program in 2012. Sergei gained considerable undergraduate research experience and exposure to NMR technology in labs both at UC Santa Barbara and UCLA. It was perhaps no surprise that Sergei should join my lab for his PhD research to further develop his NMR skills. Sergei undertook an extremely challenging research project using NMR to elucidate the structure, dynamics, and interactions of the transcription factor CREB. Given the large size of the protein and the fact that it contains both disordered and folded domains, he faced numerous challenges, but excelled at meeting them using both brute force NMR methods and INTEEN approaches to independently label individual domains of CREB with NMR active isotopes. Using these methods, Sergei obtained unprecedented insights into the mechanism by which phosphorylation of the disordered regions of CREB functions as a rheostat to regulate its DNA binding activity. Beyond his outstanding scientific contributions, Sergei will always be remembered for two additional attributes. In what is an excellent trait for a scientist, Sergei was the most inquisitive student I have ever met. He had a burning curiosity to know what lay behind every closed door. Early in his tenure in my lab, Sergei also revealed his passion for computing and data analysis. After taking one look at my outdated lab website, he immediately volunteered to create a new, attractive, and highly functional one. He also acted as webmaster for the Society of Postdoctoral Fellows at Scripps, redesigning the website to improve its functionality. Given his interest in data analysis, it's perhaps not surprising that Sergei decided to make it his career and has joined the local company as a data engineer. Sergei, it was a great pleasure to work with you. I'm delighted to congratulate you on your achievements, to thank you for your many contributions to our lab, and to wish you success in your future career.
One of the most rewarding things in grad school was conducting research to address and answer previously unanswered questions and further our understanding of things. To that end, I want to first thank my advisor, Peter Wright, for having me in his lab and his support throughout grad school. I also want to thank my committee members, Jane Dyson, my committee chair, Ian Wilson, Ashok Dennis, and my external committee member, Mark Montmany, for their support and advice. I also want to thank the graduate office and administration staff at Scripps. Finally, a big thanks goes to my family that supported and helped me throughout my time in grad school.
I have great pleasure in congratulating Ke Yang on attaining the degree of Doctor of Philosophy. Ke is originally from Inner Mongolia in China. She joined the Scripps graduate program in 2014 after completing a Bachelor of Science degree in chemical biology at Tsinghua University in Beijing. Before joining our graduate program, Ke gained considerable research experience as an undergraduate, working in labs at Tsinghua University, as well as at the Indiana University School of Medicine. Ke joined my lab as a rotation student in 2015. She was initially a little hesitant and concerned that NMR was too difficult and too challenging. Fortunately, Ke accepted that challenge and rapidly mastered the underlying technology, performing and analyzing complex NMR relaxation experiments to probe the kinetics of protein-protein interactions, which eventually led her to publish a detailed protocol to guide other researchers. Ke's thesis research focused on structural characterization of a human T-cell leukemia virus protein named HBZ, and she also analyzed its interactions with a key cellular protein named CBP. Using NMR, X-ray crystallography, and biochemical methods, Ke obtained unprecedented molecular insights into the mechanism by which HBZ deregulates pathways of blood cell differentiation and promotes T-cell proliferation and leukemia. In addition to her love of science, Ke has a passion for and a very deep understanding of music. She's an excellent pianist, as I can testify, having heard her playing the piano at a graduate student retreat. Since completing her PhD, Ke has taken up a new position as a scientific analyst at a patent law firm in Del Mar. Ke, it was a great pleasure to work with you for the past few years. I'm delighted to congratulate you on your achievements, to thank you for your contributions to our research, and to wish you every success in your future career.
Hello everyone, my name is Ke Yang. I am graduating from Dr. Peter Wright's lab this year. First of all, I would like to say a huge thank you to Peter and Jane for their guidance and support and for making the lab such a nourishing environment for my growth as a graduate student. I would like to thank Dr. Ian Wilson for his help and for our collaborations. Thank you very much. And I would like to thank Dr. Asher Dennis for his input in my thesis and for serving as my committee member. I would like to thank everyone in the Wright Lab for making the lab such a fun place to work at. I've learned a lot from all of you. And lastly, I would like to thank my friends and family on 3 different continents and 7 different countries. I know we don't see each other often, but your love and support and faith in me has always been with me, has always been felt. And thank you all for your love and support.
I'm Jin Hain Zhang from the chemistry department on La Jolla campus. Congratulations, Dr. Yao Xiao. Yao Xiao received his bachelor's degree in chemistry from Cornell University in 2015, where he conducted research in supramolecular catalysis under the supervision of Professor Chad Lewis. He then joined the Scripps. In my laboratory, he developed novel transient dividing groups and new ligands for palladium-catalyzed diverse sp3-CH activation reactions of free amines. Yao Xiao will join the lab of Professor Craig Hawkins at UC Santa Barbara for his postdoctoral studies on polymer chemistry. Congratulations, Yao Xiao. Congratulations, Dr. Hojun Park. Hojun was born in Swan City and grew up in several different cities around the world, including US and China. He received a bachelor's degree in chemistry from Seoul National University as the recipient of the most prestigious presidential science scholarship. During his undergraduate studies, he conducted research with Professor Cho-Bon Lee on transition metal catalysis. He then joined my laboratory as a KFAS fellow. And during his PhD studies, he focused on exploring new dividing groups for palladium chemistry and discovery of new ligands. And recently, he started a postdoc position with Professor Steve Buckworth at MIT to study copper chemistry. Congratulations, Hojun. Congratulations, Dr. Kita Tanaka. Kita grew up in Osaka, Japan, and received a bachelor's degree in chemistry from Osaka University, where he learned the importance of synthetic chemistry and biochemistry. He then joined my laboratory to develop a useful synthetic methodology. During his PhD study, he made several very important contributions to the area of remote CH functionalization reactions, which allowed a rapid modification of biologically important, very complex molecules. In his private life, he also managed to keep a remote relationship with his girlfriend in Japan, and recently got married with her. And very recently, he joined MIT in Professor Chris Cummings' lab to study inorganic chemistry. Congratulations, Peter.
Through my PhD study, I could learn how to find important problems and how to tackle them in the field of organic synthesis. I could not go through this long journey without the help from many people. I'd like to especially thank Professor Jin Hain Zhang for giving me a chance to work in his lab. His enthusiasm to the chemistry attracted many talented postdocs and students, which made a highly productive research environment. I also would like to thank my family, especially my parents, who are always supportive of what I want to do. I also want to thank my wife, who always cared my health and encouraged me to pursue my dream.
Congratulations, Dr. Prisa Verma. Prisa grew up in India and received a bachelor's degree as well as a master's degree from IT, where she studied computational chemistry. She then had a brief visit in Japan in Tokyo University to study transition metal catalysis before he joined my laboratory. During her stay in my laboratory, she not only did many computational studies on our reactions, but also independently discovered a iridium-catalyzed sp3-CH functionalizations. Recently, she joined MIT to study AI-based chemistry. Congratulations, Prisa.
I will now confer the doctoral degree upon the candidates. Will all doctoral candidates please rise wherever you are? By the authority vested in me by the Board of Directors of the Scripps Research Institute, and on the recommendation of the faculty of the Skaggs Graduate School in Chemical and Biological Sciences, I hereby confer upon each of you the degree of Doctor of Philosophy. Graduates, please move your tassels to the left side of your mortarboard. Congratulations to all.
So that's a wrap for our virtual graduation. So please join me in thanking and congratulating all of the graduates from the class of 2020. It's been a special year.